I'm going to teach you about membrane receptors. Here we have the cell membrane. The cell membrane is made up of phospholipids. Here you have the heads, which are hydrophilic, and you have the tails, which are hydrophobic. Within the cell membrane, you have a membrane receptor. A receptor has a receptor site here, which small molecules called ligands on the outside of the cell binds to the activation site and activates the G proteins. There are many different types of ligands that can activate a G protein, such as fatty acids, proteins, peptides, or amino acids. On the inside of the cell, you have the group of G proteins. The G proteins are made up of 3 different subunits, one being gamma, the other being beta, and alpha. Binded to the alpha subunit, you have GDP, which is short for guanosine diphosphate. Now, when a ligand binds to the receptor site of the membrane receptor, the G proteins change conformation, which means the GDP detaches from the alpha subunit and GTP, which is short for guanosine triphosphate, attaches to the alpha subunit. Guanosine triphosphate attaches to the alpha subunit. It then activates the alpha subunit, which then separates from the gamma and the beta subunit. This process can be repeated as long as the ligand stays connected to the receptor site. Once guanosine triphosphate is binded to the alpha subunit, it then binds to the activation site of adenylyl cyclase. Once adenylyl cyclase is activated, it catalyzes the conversion of ATP into cyclic AMP, which is short for cyclic adenosine monophosphate. Cyclic AMP is a second messenger used for intracellular signal transduction. Once ATP converts 4 cyclic AMPs, it activates other molecules such as pKa's, which is short for protein kinase A, which is the binding site of the regulatory subunit. This allows the catalytic subunit of the pKa to dissociate, and the pKa can then phosphorylate intracellular targets. For proper cell function, the cell must be able to stop the G protein signaling pathway after it has accomplished its task. To stop the signal, the cyclic AMP must be broken down using the enzyme cyclic AMP phosphodiesterase. The catalytic subunit of pKa then reassociates with the regulatory subunit. In order for the G protein to become inactivated, the alpha subunit must hydrolyze its bond with GTP back to GDP using its GTPase activity. The alpha subunit then reassociates with the beta-gamma complex. The G proteins are then back in an inactive state. Then the process repeats itself and the cell is ready to be stimulated by another hormone. Thanks for watching.